Hey, this is Mrs. Dellosso, and in this podcast, we are going to be talking about protein synthesis. In other words, we're going to be talking about how cells actually manufacture proteins. So, with this process, we're first going to start with DNA. And I want you to think of DNA as a blueprint. So, what's a blueprint? A blueprint are like a set of instructions in order to make something. For example, you have blueprints to make buildings and houses. So DNA is like the main original blueprint, and it's the blueprint of life. But when we think of DNA and we think of it as a blueprint and the original blueprint, it's very, very important. You know, sometimes we don't want to give our original out because it could be lost somewhere, someone could misplace it, and that won't be good because the blueprints are very important. So instead of sending DNA out of the nucleus in order to make the protein, we are going to make a copy of this blueprint, and this copy is going to be called messenger RNA. Now, this process of making a copy of DNA in the form of mRNA is called transcription, and it occurs in the nucleus of the cell. All right, so now we have the instructions for building something. Now, where is it going to be assembled? In terms of the protein, we have the message or the code in the form of mRNA, and now we needed to get it to the place where it's going to be assembled into the protein. So where does this actually happen? This will occur at the ribosome, and in this example, since we're talking about blueprints, it will be the factory. So the ribosome is known as the protein factory. This occurs outside in the cytoplasm and in the ribosome, and this process of translating the code from messenger RNA into the protein is called translation. Let's take a look at this in a different way. So we have protein synthesis that's going to be divided up into two steps. The first step is called transcription. This is where we transcribe or copy the code of DNA into messenger RNA. The second step is translation. This is when we're going to translate the messenger RNA code into amino acids. And what's the result of this process? We'll form a new protein like an enzyme, an antibody, a cell receptor, or even a hormone. So let's take a look at some of the types of RNA that are going to be involved in the process. The first type that we're going to take a look at is messenger RNA. So messenger RNA, like we just mentioned before, is made from DNA, and it contains the code to make a chain of amino acids or a polypeptide. Remember, the chain of amino acids is also known as a polypeptide. So it's going to carry the code from DNA in the nucleus to the ribosome. And the ribosomes are located in the cytoplasm of the cell, and the ribosome is going to be responsible for reading this code. So ribosomal RNA, or rRNA, it just combines with various proteins in the cytoplasm to form a ribosome. And then we have transfer RNA, or also known as tRNA. And this is important because the tRNA contains something called an anticodon. And this anticodon has the complementary base pairs for specific codons for the messenger RNA. So what does that mean? So let's say, for example, on our messenger RNA, we have a codon AUG. So a codon is 3 bases of RNA. 3 bases, we're talking about RNA. So transfer RNA is going to have the anticodon. So in other words, if I were to bring this tRNA over here, it's going to have the complementary base pair. So following our base pair rules, our A would pair with U, remember, because we're talking about a type of RNA. Our U would be paired up with A, and our G would be paired up with a C. So this would be tRNA's anticodon. And this is going to be important when we talk about the process of translation. But here are the 3 types of RNA. Okay, so the first step that we talked about in the process of making a protein is called transcription. Remember, that's making messenger RNA from this DNA. So let's take a look at what that looks like in more detail. So in the process of transcription, we have this important enzyme called RNA polymerase. And what RNA polymerase is going to do, and this is RNA polymerase is this gray structure here, that enzyme, it's going to be responsible for unzipping the DNA and then adding the RNA nucleotides based upon that DNA. So this blue strand here, right, both of these represents DNA, and if you notice, the enzyme has untwisted, it's separated two of the strands so that the messenger RNA nucleotides can be added. So where we're taking it from, here we have a T in DNA, so the A nucleotide will be added, we have an A, then a U nucleotide will be added, and this will form that messenger RNA. Now, this first process, this is going to form something called the pre-mRNA. Let's take a look. So here's our DNA. From the process of transcription, we're starting to make the pre-messenger RNA here. And from that, then we'll make the messenger RNA that will leave the nucleus to the ribosome. So what does the pre-messenger RNA look like, and what does it entail? So here's our strand of the pre-messenger RNA. And the pre-messenger RNA contains what's called exons and introns. Now these introns don't actually code for amino acids, so then enzymes will actually snip these introns out, the exons will be joined together, and now we have our finished messenger RNA molecule, which will now be ready to be translated into the protein. So looking at our cell once again, now that we've snipped out the introns and have our messenger RNA, the messenger RNA is now going to leave through a pore in the nucleus and travel to the ribosome, where it will then be translated into the polypeptide, or the chains of amino acid, which will then be formed into the protein. So let's take a look at translation in more detail. All right, looking at number 1 here, here is our ribosome, and you can see going through the ribosome is the messenger RNA. So now, what's going to bring the amino acids so that they're joined together? That's where we have tRNA. So the tRNA anticodon aligns with the messenger RNA codon. So here's the tRNA, see how it almost looks like a T? It's going to bring the anticodon, which will match up with the messenger RNA, and it's also going to bring the appropriate amino acid. Take a look at step 2 here. The chain of amino acids is added to the amino acid brought in by the tRNA. So now this whole chain of amino acids that has been brought over is now going to be added to this one, elongating the amino acid chain. Now, step 3. Now, once that tRNA has done its job, it's then going to leave, will be released. Number 4, another tRNA is going to bring in a new amino acid as the mRNA shifts. So let's take a look at that in action. I have a nice video clip to show you of translation. Okay, so here you can see the tRNA with the amino acid on top. Another tRNA has joined at the ribosome. Then the amino acid will be joined to the new one. Here we have new tRNAs coming in. And now we can see how the amino acid chain is being elongated. tRNAs keep coming in, bringing amino acids. And then eventually, once it reaches the stop codon, the translation process will end, and the amino acid chain will then be released to form the protein. So the next thing that we need to look at is, well, what amino acids do the messenger RNA code for? So here's our strand of messenger RNA, and what I want to do is break it out into its codon. So that means every 3 bases will be a codon. So our first codon is AUG, followed by the next codon CAU, and so on. So here are all of our codons. So each of these codons of messenger RNA codes specifically for amino acid. Now here's how you use the chart. So here's looking at AUG. A is our first base, U is the second base, and the G is the third base. So when you look at a chart like this, you're going to look on the gray part here, and you'll see where it says first base, second base, and third base. So our first base, remember we said, is A. So I'm going to find my A over here. My second base is a U, so I'm going to look on top for my second base, and I'm going to scroll down to the section where the first A was. Now the next, or the third base, is a G, so I'm going to look for the G within the section that the first two bases are. So for example, here's my G. I'm going to look over. So my G is here, and if I stretch my line, whoop, real quick over here, that's going to be methionine or met. So my first amino acid is going to be met. I'll do the second one again with you. So here's my codon, CAU. I'm going to look for C, my first base. My second base is A, so I know if I look for where these two meet, it's going to be somewhere in this region. And the third base is U. So if I look for U here and scroll over, the second base will be the second amino acid will be HIS. So try and do these last 5 codons on your own using this chart. You can also see on this particular chart, if you look for the codon, the letters, it's there, but sometimes they don't have that on different charts, so you'll have to use this way to find it. Alright, so that's the overview of protein synthesis, and I hope that you found this helpful.